Just a reminder, everyone, that if you want to get documentation of your continuing education credits, you should contact Dr. Eric Seagren, and he will provide that documentation for you. Also, just a reminder, or for those of you who are just joining the Environmental Engineering Graduate Seminar Series for the first time this semester, we do have all participants on mute and unable to share their video. If you have a question for our speaker today, it'd be very efficient to type it into the chat, or please raise your hand at the end of his seminar, and we'll try to get to as many of those questions as we can. It gives me really great pleasure to introduce today's speaker, Professor Bruce Rittmann, who is the Regents Professor of Environmental Engineering and the Director of the Biodesign Swette Center for Environmental Biotechnology at Arizona State University. Professor Rittmann's research focuses on the science and engineering needed to manage microbial community to provide services to our society. And these services include the provision of renewable energy, the cleaning of water and soil, and improving human health. For his body of work in this area, Professor Rittmann has received just about every recognition and award possible. He is a member of the National Academy of Engineering, a fellow of the American Academy of Arts and Sciences, the Water Environment Federation, International Water Association, the Association of Environmental Engineering and Science Professors, and the National Academy of Inventors. And he's a distinguished member of the American Society of Civil Engineering. Dr. Rittmann was also awarded the inaugural Clark Prize for Outstanding Achievements in Water Science and Technology from the National Water Research Institute, and he has received the Walter Huber Research Prize and the Simon Fries Award from the American Society of Civil Engineers. Professor Rittmann is also the GM Fair Award recipient for the American Academy of Environmental Engineering and Sciences, and he is the recipient of the Perry L. McCarty AESP Founders Award. He also is the co-winner of the 2018 Stockholm Water Prize and has authored many articles and books, including the textbook Environmental Biotechnology Principles and Applications, which he co-authored with Professor Perry McCarty. On a personal note, I was also very fortunate to have Professor Rittmann as one of my 2 PhD advisors, and his mentorship and the classes that I took with Professor Rittmann not only shaped my interests, but really had a profound impact on my own research, the way that I teach, and certainly the way that I write. So this has been a great experience for me, and I will turn it over to Professor Rittmann without any further ado.
Great.
Thanks.
Thank you, Jennifer. I'm always proud to claim Professor Becker as one of my PhD students, and also Professor Seagren. So I have a great connection here to the faculty at Michigan Tech. By the way, I've been at Arizona State University now for just over 16 years, and I decided to go to Arizona State University in the fall of 2004, and I did this like the day before I embarked on a AESP lecture tour, distinguished lecture tour, and my first stop was Michigan Tech. So the people at Michigan Tech were actually the first people to know that I was moving to Arizona State University. So lots of interesting connections here. Alrighty, so today I'm going to talk about microorganisms closing mass balances. This is one of my favorite topics, and I'm glad that Professor Becker selected this one for my talk today. Okay, so let's see here. So first of all, a little bit of context. So one of the things that's really important in our field right now is a whole range of molecular biology tools, the omics, and I've made a collage of a bunch of them here that we use. and they're all really great tools on this. And I'm really happy to say that back around in the early 1980s, I was really the first environmental engineering professor, maybe I guess first environmental engineer to get the opportunity to use these tools. And we at that point had RNA hybridization, extraction and hybridization, and that was it back in the early 80s. Now we've got all these other ones and a bunch of other ones that I couldn't fit on my collage. So these tools have become in widespread use. They're really valuable and very powerful. And because of that, well, you can use them in combinations, but because of that, they're really fabulous and popular. You can see, look at the enthusiasm of these people here for these omics tools. And it's really quite amazing. Let's see. I gotta make sure I learn how to use the forward button. Okay, so these omics tools are really fabulous, but there's a but here. And the but is they're not really truly quantitative. So if you're an engineer like me and most of you, you might like to quantify things. You might like to do mass balances, and these tools don't really quite do that for us. For example, you'd like to quantify how many different types of a microorganism there using the 16S ribosomal RNA gene. But the number of operons for 16S RNA are highly variable, somewhere between 1 and 15. And here's some data on that. A paper by Vitrovsky and Baldrian, who tabulated in 2013 the number of RNA operons in different bacteria. And you can see in this graph, they range from 1. A lot of them have only 1, but even more have 2, and then 3, and then 4 and 5, and so on, up to one of them has 15. So it's really hard to do quantification of biomass targeted to the 16S ribosomal RNA gene because they have so many of them. And this is a real challenge for us in doing engineering stuff, and that engineering stuff is usually quantitative stuff. So one of the big mistakes we can make when working in microbial ecology is to rely too much on these tools here. They're very great, but we shouldn't ask them to do things that they can't do very well. And if you ask them to do something they don't do well, you might get an answer back that causes you to fool yourself. You might fool yourself about what's important. And the information actually might be very misleading, at least if you apply it in a very blind, naive way. And that's what I'm going to talk about today, is how not to apply these wonderful tools in a blind or naive way, but to use them in a very sophisticated way and in concert with a range of other wonderful tools that we have, more obvious engineering tools. So the key thing here is that the microorganisms themselves never get fooled. We can get fooled, but they never get fooled. And that's one of the themes of my talk today. Let's be like the microorganisms. Let's not be fooled, because the microorganisms always close the mass balances. They have to close the mass balances. This is how they live. And so they close mass balances on things like electron equivalents, nutrient equivalents, acid-base equivalents. Now in my talk today, I'm going to talk about electron equivalents a lot and nutrient equivalents to some degree. I won't have time to talk about acid-base equivalents today, but the principles apply to acid-base as well as these other ones. So let's get started. And to get started here, I'm going to put up this diagram, which is an adaptation from what's in my textbook, Environmental Biotechnology here. And it's basically an electron flow balance. Where do microorganisms live? By moving electrons around. This is probably the most fundamental thing we can remember about microbial systems and any type of microorganism. So they have some electron donors. Substrate shown here at the left, it'll get oxidized. It'll lose its electrons. Some of those electrons will take the top path and go to some acceptor, and this will generate energy. The squiggly line here is the generation of energy. The organisms will then take that energy and combine it with the rest of the electrons to form biomass, and they're living. This is the main thing that microorganisms do. And they extract electrons from the donor, they gain energy, they use an energy and other electrons to create biomass. This is the core of life. And whether it's a single organism or a bunch of organisms, this is what goes on. But of course, in microbial communities, it's not just one organism. It's one organism and another organism doing something similar, perhaps with the same substrates, perhaps different substrates, but the same basic pattern. And there's usually another one and another one. And of course, I could keep going on like this because there are lots of microorganisms that are out there doing this basic process of electron transfer and capturing electrons and energy into their new biomass on this. So they're all doing it, and we, to understand them, have to understand how they're doing it and actually quantify how they're doing it. So I'm going to give two examples here where we're going to combine together this concept of quantification and mass balances with some of the power of the molecular tools. So two examples that are going to illustrate the linkage, really good and fruitful linkage of these two kinds of tools. So first one is one, this was published, and if you look down at the bottom, you'll see that it was published in 1999. So this is more than 20 years ago. So this was based on work that was done in the late 1990s. And this is the paper that we published on it. There was some really good engineering quantification work done by Chrissy Laspadu. She's underlined, and some really great work done on the molecular biology side of it. That was by Jody Flack. She's underlined there too. And we had a big team. So it was the late 1990s. And our team, then a team at Northwestern University, had just developed the first set of oligonucleotide probes for identifying ribosomal RNA from all known nitrifying bacteria, ammonia oxidizing bacteria, and also the nitrite oxidizing bacteria. These are all the ones that our field knew at the time in the 1990s. We know more now, but at the time, this was all of them. And we were working and funded by a private company called Société Lyonnaise des Eaux at the time. It's now called Suez. And Suez is one of the world's largest environmental companies. They operate water and wastewater treatment plants around the world. They do it now, and they did it back in the 1990s. So they were funding our research because they were very interested in the reliability of nitrification in their wastewater treatment plants. So they funded our work to develop and use these probes. They had some treatment plants in France and in the Netherlands. And what we decided to do, with their partnering, was to try out our exciting new oligonucleotide probes with the biomass from those treatment plants. This is really great. It's a great opportunity. We don't often get to do something so practical and fundamental at the same time. So they sent us mixed liquor samples from all the treatment plants, and then we did our oligonucleotide probing, looking at all the nitrifiers and a few other things that I'll talk about as we go on. They also sent us the data on their process operation. And you'll see the word limited here in red. If you've ever worked with treatment plants, you realize that they don't really collect a lot of data that's immediately relevant to understanding microbial ecology. But we got everything they had. And then we knew, because our probes were so wonderful, that we would gain tremendous insight into the operation of these treatment plants from looking at the results of our probes. So that's what we expected, and that's what our sponsors there at Lyonnaise des Eaux expected as well, I should say. So what was the reality of it? We did all the probing, we got the data, and the result was nothing made any sense at all. Nothing. It looked like complete chaos of the data. In fact, one could imagine that if you had used a random number generator, you could have gotten data equally sensible as we had there. Well, I can tell you that this lack of any obvious order or cause and effect in our oligonucleotide probing data was distressing to us, and it was very distressing to our funders and partners who were expecting something quite amazing to come from it. And so it's created a little tension in the team here. So here's an example of kind of results we had. This is from the paper, one of the tables from the paper. And you can see on the left side, we had the 1, 2, 3, 4, 5, 6, 7 plants from France and the 2 plants from the Netherlands. And you can see the kind of information that was available from operations, the volume, the HRT, the BOD loading, and the influent and effluent concentrations, mixed liquor, volatile suspended solids. And the right-hand column is saying whether or not there was any obvious evidence of nitrification. And obvious evidence of nitrification would be the formation of nitrate and nitrite. And you see some yeses and some nos. Now, the interesting thing about this was that you could see here, for the ones that I put the circles around, there's no evidence of nitrification yet. Yet, when you look at the influent versus effluent TKN, total Kaldol nitrogen, all of the reduced ammonia level nitrogen, there was huge removals. And so the question was, gee, how could we be getting all these huge removals of nitrogen, TKN nitrogen, when there wasn't any nitrification? Well, this was really concerning, intellectually concerning to us, and particularly concerning since the operators of these plants often believed that they were actually getting nitrification. So what do we do about this? Wait a minute. I forgot one more piece of information. We did, of course, our oligonucleotide probing for ammonia oxidizing bacteria and also general all bacteria. And we did ratios of ammonia oxidizing RNA to VSS ribosomal RNA, and same for total bacteria. And the ranges were huge. Ranges were huge. You can see the ranges I've listed there. 66 to 570 nanograms of RNA per milligram VSS for the ammonia oxidizers, and similar for the other. And so what we had were these huge ranges, and they didn't seem to correlate in any way to any of these data. It was really a mystery, a real mystery. So now I even knew back in 1990s that the microorganisms always close the mass balance. So I insisted that our team develop a simple mathematical model that would do 2 things. One, would use only the data we actually had, the kind of data I just showed you on that table a couple of slides back. And we could systematically solve mass balances for BOD and nitrogen, and taking into account the competition for nitrogen between heterotrophs. Heterotrophs use nitrogen strictly for synthesis, and the nitrifiers, who mainly use nitrogen as their electron donor in respiration. And so there were 2 different uses for nitrogen, and we needed to be able to take those into account. So back to our paper. So we published this paper, and the person who did all the hard work on this was Chrissy Laspidou, then a PhD student. She's now a professor in Greece. And she developed a series of equations that I'm going to show you here. If you go to my textbook, you'll see that these are the same kind of equations that we have in the textbook. It's not exactly the same as this, but these are all the same kind of equations. And so let's take a look at this first equation here. On the left side is XV. This is the mixed liquor volatile suspended solids. It's a number we knew. We had a measurement for it. The next thing on the right side of the equal signs is theta X over theta, the solids retention time over the hydraulic retention time. We knew that HRT, the theta value, so we had that. There are a bunch of other parameters in here, like delta BOD and delta TKN. We actually had those numbers here. They were, particularly delta BOD was measured in, at the treatment plant. And we could do some calculations to get delta TKN on that. And then you see there's a bunch of other parameters, things like 1 minus F sub D. F sub D is the fraction of electrons, the fraction of the biomass that can be decayed through endogenous respiration. And B is the decay coefficient, and theta X is in there. There's also 2 Y values, the yield coefficients for the nitrifiers and the heterotrols. Well, most of those values, most of those parameters have values that we know pretty well. And so some of those values in there were either measured at the plant or could be estimated from what we know about how microorganisms work. The big parameter here that was not known was theta X, solids retention time. They didn't measure it at the treatment plants. They didn't report it to us, but we could use this set of observed values and some knowledge about microbiology to calculate theta X. So we use this equation here to compute the solids retention time, or theta X. So now we actually had a measure of theta X. So this was a great thing for us. Now, armed with theta X, we could then make 2 other calculations that were ratios of the amount of active biomass that were the ammonia oxidizers, that's the left-hand side, or the ratio of the amount of active biomass of the heterotrophs on the right-hand side, both of them normalized to the total mixed liquor volatile suspended solids. And you could see that both of them have theta X over theta in the numerator, so we needed to know that theta X. And the one on the left just tells us about the removal of TKN by nitrification. So that's information we could glean from the previous page. And then on the one on the right, we had mainly biomass produced by BOD oxidation. So once we got theta X, we could then go in, again with plant data and some knowledge of things like yields, and we can compute the mass balance generated ratios of the ammonia oxidizers to total biomass and the heterotrophic bacteria to total biomass. So, or actually it's total, the one on the right is actually all active biomass, but it's mostly heterotrophs. Okay, so we did that. And then we started to look at the data again with new eyes and with new information. And so this is a table that has some of the information from before, and it has some new information that we generated from our molecular techniques and from our mass balance analysis. So again, you can see the left-hand side, the 9 processes, and then pretty soon you see the theta X values, the solids retention times, and there's quite a range there. One is as low as 0.42 days. Wow. And then some of them are up, almost 100 days. And so there was a huge range in solids retention time. Now, if you span over here and you look at the model translated results, the model translated results tell us that the 3 plants that had really short theta X values, and they're the ones that are circled, also had model translated results of no ammonia oxidizing bacteria. And by the way, they're also the ones where we didn't have any evidence, direct evidence of nitrification in the system. So in those 3 cases, we didn't have any nitrifiers and we didn't have any nitrification. And the reason, the solids retention time is just way too short. Now, all the other ones had substantially long solids retention times, and they could have had and should have had nitrification. And you can see again on the right-hand side, there are ratios of how many ammonia oxidizers there were to the biomass in 2 columns there, different ratios, but the same information. And then there's also one on the very far right, which is the amount of active biomass total to the total mixed liquor volatile suspended solids. All right, so just looking at this table, we could explain why it was that we had, oh, wait a minute, I forgot something. I forgot to look at the middle of the table, sorry. At the middle of the table, you will see our molecular probing results, and these are expressed as ratios of different types of organisms to each other, or different types of organisms to total VSS expressed as RNA. And you have here some interesting numbers here. In the ones that are circled, you see some very low ratios. I'm looking at the XAO for XA for the ammonia oxidizers and XA for the heterotrophs. They're quite low in the circled ones. Also are the absolute values of the probe results to VSS. So in all those cases where we didn't have any nitrifiers, we saw some response, but it was low. And we figured we figured out that this was just some kind of a methodological thing. There's some background level on that probe. Okay, so now let's go on to interpreting this a little more. So here is a plot of the ratio of ammonia oxidizers to heterotrophs. And on the horizontal axis, this is the model predicted result. And on the vertical axis is the RNA probing results when we use just the organisms that had nitrifiers. And you can see, by the way, the dark line, that's a vertical line, is the 1-to-1 ratio, or almost 1-to-1 ratio. And what you can see is that we have a really good correlation. That is, the molecular probing data actually represented quite well what the mass balance said had to be the ratio of nitrifiers to heterotrophic bacteria. It was really quite good once you had nitrifiers. And so this was really quite good. And as you can see with that blue arrow here, as we increase the TKN to BOD ratio, that is, you put more nitrogen in the system, you got more nitrifiers. Well, this makes complete sense. You put in more nitrogen, you should have more nitrifiers to do the job. And here's another plot here where we're looking at now all bacteria. We used the EUB 338 probe, which was for all known bacteria. And we plotted that, the probe results on the horizontal axis, and the model predicted a ratio of active biomass to VSS on the vertical axis. And you can see, again, very, very strong correlation here of the results. They did very well. And the other thing you can notice is that there's a blue arrow. And as we go from right to left, we have increasing solids retention time. And so the ones that are way off to the left have a very long solids retention time, lots of decay, and you end up with rather few active organisms and a lot of other stuff in there. So again, that makes complete sense. And the probing results really agreed very, very well with the mass balance results reflected through the model. So we actually made complete sense of everything. Everything agreed. The microorganisms did the mass balance, and when we did the mass balance, we understood what was going on and why. So combining all of this together here, we found that when we didn't have TKN removal, you had no nitrification, there was still a little background signal for the ammonia oxidizers, and we had to subtract that out because the nitrifiers really were washed out. Once we had nitrification, then the ratio of ammonia oxidizers to total biomass was controlled by the TKN to BOD ratio. More TKN going in, more nitrifiers. The ratio of active biomass was inversely proportional to the solids retention time. It should be. A long solids retention time has a lot of decay, and you build up a lot of inert biomass. In short, everything in the treatment plants worked like it should. The molecular results, the mass balance modeling approach gave completely consistent results, and everything was logical. And I really felt super great about this work, because we really did make sense out of what appeared to be chaos. And we did it by combining mass balances with our molecular probing results. Okay, so that was my first example. And now I'm going to give a second example, a much more contemporary example. This is a paper that was published in Biotechnology and Bioengineering, 2019. So 20 years later from the first paper, we're doing a second paper where we're going to apply similar principles, but to a very different setting. So let's go on to this one here on bubbleless oxygen delivery and aerobic benzene activation, followed by denitrification. Oh, by the way, the person who really was the leader on this was Dr. Chen Zhou's research professor with me in the Swette Center. All right, so here's a little diagram of what goes on through normal aerobic oxidation of benzene. Okay, so there's benzene off to the left. You can see it's a ring with no substituents. It will get oxidized by either a monooxygenation, two monooxygenation reactions, or one dioxygenation reactions. The key thing about these reactions is that they require molecular oxygen. They require molecular oxygen, and you can see it right here in the diagram as I show it. And when these one or two reactions occur, they produce catechol. And you can recognize catechol. It's the benzene ring with two OH groups. Then the catechol is cleaved, broken into two parts by another dioxygenase, use more oxygen, and then it produces products that can be oxidized by the typical metabolism of hydroxylations and dehydrogenations on this. So that's mineralization. But the key thing for aerobic degradation of benzene is you have to get these initial steps that require molecular oxygen. They absolutely have to have it. And so this was our starting point on it. Now this is great, except that when you oxidize benzene completely using oxygen, not only for these initial reactions, but also for the respiration to mineralize it, it consumes a lot of oxygen. And this is expensive to supply. It takes a lot of energy and therefore a lot of money to supply this oxygen. Besides, when you're dealing with a compound like benzene, which is volatile, you can actually strip off the benzene instead of biodegrading it. So there are lots of reasons why you just don't want to be blasting your biological reactor with a bunch of oxygen. So these were our sort of our starting considerations on it. So we can, oh, by the way, you can also do anoxic benzene oxidation. That is, you don't need any oxygen for those first steps, but it's very slow. And so we didn't really want to consider that. So what we did want to consider was what we call a hybrid process, And in a hybrid process, we're going to do this in two stages. In stage one, we are going to use oxygen to do those initial monooxidation and dioxygenation reactions to make catechol. Catechol, again, is C6H6O4. And then we're going to take the catechol and we're going to mineralize it and do respiration using another electron acceptor, one that is not oxygen. And we chose nitrate as our acceptor in this case. So, and what you can see here is that we're going to save about 60% of the oxygen delivery demand if we use something else in the respiration step. Besides which, we're really minimizing the need to add so much oxygen and reduces the risk of volatilization. Okay, so we did this in the oxygen-based membrane biofilm reactor. Because the membrane biofilm reactor in general is one of my long-standing research and development areas. And in general, what you do is you grow a biofilm on the outside of gas transfer membranes. And the biofilm then consumes a gaseous substrate that you deliver through the membrane wall. And then it uses that and removes whatever is in the water on the water side. Now, in the oxygen-based system, we're delivering oxygen gas to the membrane. And in this case, the benzene is in the water and it's going to be oxidized as I showed you in the earlier slide. So this is what it looks schematically. You can see there is a reactor with a bunch of membranes in it and we were delivering oxygen to it. This is what it looked like after we ran it for a while. And you can see very nicely there's a sort of a tannish biofilm that's growing on the outside of those fibers. The fibers themselves, if they have nothing on them, are very, very brilliant white. So this you can just see the biofilms here with your eyes on it. Okay, so we want to answer 3 questions. Question 1, can we actually do this hybrid thing and just have the first steps with oxygen and the rest of it with nitrate? If that's true, who is responsible for it? What bacteria are doing this very interesting job? And then if they're doing it, which of those initiation reactions are they actually using? Are they using the first dioxygenase or the first monooxygenase? So we got a lot of interesting questions that we can answer by a combination of mass balances and molecular biology. So here are the results. I'm just going to walk you through how to read the graph here. First of all, it's got time. We operated the system for about 120 days. In the middle, you can see the dark circles are the benzene influent concentration. It was pretty stable at about 100 milligrams per liter. Up here at the top is how much oxygen was supplied to the membranes. So initially, we started out with pure oxygen. That gave a very high oxygen transfer capacity. And then we moved to air. So we reduced the delivery capacity to about 1/5. And then we went down another tenfold with only 2% oxygen. The idea here was to, as we move from left to right, was to continually restrict the delivery of oxygen so that at some point there'd only be enough oxygen to do those first reactions and nothing more. Then the organisms would be forced to use nitrate, which we of course had put in the influent. You can see down here in the middle graph, the open circles are the benzene concentration. And when we gave it lots and lots of oxygen, we got essentially 100% removal. As we started to cut back on the oxygen, the removal did go down, but it stayed pretty good at about 50%. And down here are the electron, all in electron fluxes, electron milliequivalents per square meter of membrane per day, comparing the oxygen delivery capacity here in the orange dots with the surface loading of benzene, which is going across the top. And you can see at a certain point, right around day 27 or so, the delivery capacity of the oxygen was way less than was needed for complete mineralization of the benzene. So at this point, we should have seen this shift. All right, so we systematically lower the oxygen delivery. We still had about 50% removal, and we never saw any oxygen at that point in the water. Okay, so what's going on here? So you have benzene, you do the initial reactions, the mono or dioxygenations, and you use the oxygen and you create these short-term intermediates, short chain intermediates, like catechol and downstream of catechol. And then what are the organisms going to do with this? They're going to respire it. They're going to completely oxidize it and then use the electrons to grow the respiration. So will they use oxygen or will they use nitrate? Which one are they going to use to gain their energy via respiration? That was the big question. Fortunately, we were measuring everything. We were measuring the benzene and all the intermediates, and we were measuring oxygen and nitrate and other downstream reduction products of nitrate. All right, so here is a summary of the results from the operation of the reactor. So here we have their steady states. Remember, as we go from left to right, we're restricting more and more the oxygen delivery capacity. And that's shown by the little yellow-orange circles up here. You can see it was very high delivery capacity at first. By the way, up here, where I'm pointing with my cursor now, is the delivery capacity of, the delivery rate of benzene expressed in electron milliequivalents per square meter per day. And this was in the same units, the oxygen delivery. So out here, in the first phase, we had plenty of oxygen, plenty of oxygen. And then as we went on, you can see we were continually restricting the oxygen supply. Whoops, excuse me. So what happened? So if you say, look out here, you can see that the organisms used benzene for the initial reactions, the mono or dioxygenations, that's in the dark one. And then they used a bunch more electrons from there for respiration to get energy, and then that made biomass. This is the gray hashed part. Okay? So that's what they were doing there. Now, when we started cutting back on the oxygen, what happened? Well, what you see here is that this dark orange hardly changed. The organisms were still using almost the same amount of oxygen for the initial reactions, but as we go from left to right, what's going down is the light yellow tan one here, where they were doing respiration for oxygen. And we're going down and down and down and down. And in its place, what went up was nitrate respiration, nitrate up and up and up and up. So the organisms were substituting nitrate in, oxygen out for respiration. And they were generating then a lot more nitrogen-related biomass that's shown here by the crossed ones here. And so they were doing exactly what we wanted them to do. They were using oxygen only for those initial reactions, the dark orange, and they were hardly using any oxygen for the rest, but they had nitrate, and they could use nitrate, and we could see it from mass balance, just how they were doing it. So we answered the first question, could we do this hybrid processing? And the answer was yes. Second question is, well, who's doing it? So we did 16S ribosomal RNA high throughput sequencing, and we came up with a really fine distribution of this, as shown in this very colorful pie chart here. Now, the key thing here is that by the end of the processing, when we were at the end and we were really restricting oxygen, by far the dominant organism was in the Comamenidaceae, something like that. These are hard to pronounce. And it dominated. It was like 65, 70% of it. Interestingly enough, we had inoculated the system with Pseudomonas, and by the end, they were down to 0.4%, so they weren't doing a good job. Now, the thing about these Comamenidaceae is we know that they can do aerobic benzene biodegradation, and we also know that they can denitrify. So we created a condition where to really live, to respire, to grow, they had to use nitrate, and look who took over. Microorganisms that had the capability of doing just that. So it really made a lot of sense. Now, another thing we want to know is, well, for those initial reactions, which one are they using? So here's a diagram of the very well-known pathways for benzene degradation, the early steps. Now, it's hard to read, so let's blow it up and look at the important point. So in the circle is benzene, and it's going to go down to catechol. There's catechol, there's benzene. It can either go through phenol, or it can go directly this would be 1, 2 monooxygenations, or it could go this way here with a dioxygenation. And we were really interested in knowing, well, which path did it take? So we used a technique called pie crust. Pie crust is a technique that allows us to examine the known functional genes in sequenced bacteria and see which ones are present based on the distribution of the various bacteria. Well, this one was dominated by this Comamenidaceae. Okay, I'm not going to say that anymore. It's too hard. That organism has monooxygenases, and it goes through phenol and then the catechol and then downstream. So we also could see from our molecular analyses, our omics, that not only was that this organism, but that they were using the monooxygenase pathway. So this was a really great insight to us. So the other thing using pie crust is we looked around to see if there were any genes for the anoxic or anaerobic activation, and we basically didn't find any. So these organisms really were doing what we wanted them to do, using oxygen for the first steps and then using nitrate for the rest. So what did we learn? We combined mass balances, we did metagenomics, and we learned a lot. First of all, we demonstrated clearly that we can control the oxygen supply rate, and we could achieve a hybrid process that would do benzene biodegradation with using oxygen only for the required initiation steps and nitrate for the respiration steps, the steps that supported energy generation and growth. We learned that this organism, whose name I'm not pronouncing anymore, was a dominant family because they could do the initial monooxygenation reactions when we gave them oxygen, and they had the capability of doing denitrification on respiration so that they could grow and thrive and actually become quite dominant in the reactor. So we demonstrated that if we really controlled the oxygen supply rate, very strict oxygen delivery management, we could get a successful hybrid process here, and we got very strong ecological selection that gave us just the organism that we wanted to do the job we wanted done. So with that, I've come to the end. I've given you two examples about how we combine mass balances with various omics techniques to gain great insight into what's going on, and also to understand better how to manage our systems to achieve the goals that we want to achieve. And with that, I'm going to close my talk. I'll be very happy to address any questions you have. Thank you very much.
Great, thank you. That was really interesting. So are there questions for Professor Rittmann? Again, if you have a question, please indicate it with a raised hand or type a question into the chat.
I'm going to stop sharing here.
Okay.
If I need to go back, I know how to get back.
Okay.
Okay.
I have a quick question with regards to sorting out those pathways. Would there have been, or did you consider also maybe using some stable isotopes or any other technique to verify the dominant pathway?
Of course, stable isotopes are very powerful for doing that, but we don't really have the capability of doing it, and so it really wasn't an option.
Sure.
In our case, you have to use what you can do on it. We were pretty happy though with the result. We think it's pretty definitive.
Yeah. Just another follow-up question while we're waiting for others to chime in. By chance, did you run a similar experiment? And I hate it when people ask me things like this, or there's always more experiments you can do, right? But had you not cranked down the oxygen delivery, do you think you would have gotten a different microbial community composition?
Yeah, I'm 100% certain we would have. And first of all, we inoculated with a Pseudomonas that was known to handle benzene. And if we'd have kept up the really high oxygen concentration, most likely that Pseudomonas would have continued to be a major player in the communities. Now, one thing about membrane biofilm reactors that we've discovered many times is that you can put in a pure culture, but you never maintain a pure culture. I mean, we operate these things for months, as you saw, 120 days there. And we never, ever maintain a pure culture in these. And so it turns out that something always gets in, and it makes it a wonderful, wonderful vehicle for doing ecological studies, because it's always ecological. I see somebody named Segrin has his hand up.
Yeah. Okay.
Bruce, great to see you, and really interesting talk. I guess you answered my first question in your last comment there. So you didn't add any other source of inocula to the second example. That was just happenstance with those organisms that got in there.
Yes. And this, we know from experience that we can put it in the purest culture we have, but it doesn't stay that way. And we're feeding these things continuously, 24 hours a day, every day of the week. you just cannot maintain sterility in every possible place to do it. So we always get this. We had a really interesting study we did a few years ago with sulfate reduction and uranium reduction. We were interested in reduction of uranium, and we knew sulfate reducers could do it. So we inoculated with we thought was a pure culture of desulfo vibrio vulgaris, and we operated it, and very quickly we got sulfate reduction, and then we got uranium reduction. It was a huge success, and then at the end of the experiment, like we did here, we went in to establish what was the microbial community, and we found absolutely zero desulfo vibrio vulgaris. It was gone. So
Yeah,
this
Yeah.
always happens. Yeah.
it's almost impossible. Yeah. Well, and then in your first case, it was interesting here too, that the combination of mass balance and the microbial ecology analysis, but couldn't in that first case you possibly have come to the same conclusion just based on conventional measurements of sludge age in those systems, if you made the right measurements?
Yes, if you had made the right measurements, you put your finger on the right thing, is they didn't make the right measurements. And when I said early in my talk is that they sent us all of the parentheses limited on parentheses results data they had. That was the message. I mean, the truth is people don't measure the right things, and so when you go into a real treatment plant situation, you end up with very incomplete information. So of course, if we had all the right information, we could have calculated the solids retention time off the bat, and we would have said, well, that's got a solids retention time by half a day. There's no way you've got nitrifiers. But they didn't have that. They didn't know that. And we didn't know it until we went through all the calculations on it. So we used what we had, and it gave us the information we needed. Theoretically were other ways of getting that information, but they weren't available to us because the operators didn't collect the data.
Right. Well, they should have read your book and the critical role of SRT and
they should have, yes. You're completely correct.
All right. Thanks, Bruce. I really enjoyed it. Thanks.
We have a couple more hands up. We'll go to Dr. Urban and then Enid, and we have a question in the chat, which I will read in a moment, but let's go to Dr. Urban.
Okay.
Bruce, I enjoyed your talk very much. It seemed to, your last example in the reactor worked well because you had, and correct me if I'm incorrect here, had essentially a single substrate, and you could follow the electron acceptors, reductors, and there weren't really the possibility for accumulation of intermediate oxidation states. In the environment, that's always much harder. In a sediment environment, we can get sulfate reduction, but then it can be reoxidized by iron oxides in the sediment. And so this following the electron balance just becomes much more difficult. And I wonder if you have helpful suggestions for those circumstances.
Yes, I mean, you put your finger on, a big challenge of the real world, and that is you don't just have one acceptor, you don't have one donor. And so, so the first lesson to learn when you consider applying this to the real world is you need to measure all the potential acceptors. Now, we only had oxygen and nitrate in that case, but what if we'd had sulfate? That would be the most obvious one. And, and if we really cut back the oxygen, almost nothing, I mean, maybe we would have even got methanogens in there. And so, yes, so when if you've got the possibility of other electron transfer reactions, you need to be measuring the things that are going to tell you what's going on in there. So, that's the number one thing. And likewise, if you're doing looking at the donors, for example, let's take a case where benzene would be important. That would be gasoline contamination, where you have BTEX, benzene, toluene, xylene, and ethyl benzene altogether. Well, now you've got four similar but not the same electron donors. And so you'd be really wise to measure each of them individually. And you can be quite certain that this would result in a more complex microbial community if you were getting, oxidation of all four of them in parallel.
Thanks.
Mhm.
Okay, I'm gonna go to the chat really quickly, and then we'll get to you, Enid. So there was a question from Adam Bell in the chat. It says, he wrote, I think you mentioned that there are two ways to move from benzene to catechol, directly and by benzene, and then through the conversion of benzene via phenol to catechol. Did you measure which path was more predominant?
Yes, well, we did that at the end by looking at what enzymes were present in the community. And we couldn't really measure catechol or phenol because they were intermediates and they were consumed. So making a direct measurement of the intermediates was just not feasible. So what we did was we looked at the community and we looked at what genes, relevant genes, are available in the community as we assess the community. And that's what PyCrest does. PyCrest looks at the complete genomes of organisms, and then you can tell it what kind of genes to look for, and it will tell you which ones are there and how many. And so we were able to see that there was a very large amount of the monooxygenase genes, the ones that go to phenol, and there are essentially nothing of the dioxygenase genes that would take it directly to catechol. So that's how we determined it. So this was a great example of using the molecular tools to supplement when the mass balance tools couldn't work. We couldn't use them because you just couldn't measure the materials, they were gone. And so now we're using the molecular tools in a very powerful way to get insights into something we just couldn't measure by chemical analysis.
Thanks. Go Bulls.
Great, thank you. And now we have a question from Enid, and this will likely be our last question.
Okay, Enid, make it a good one then.
First of all, I definitely really enjoyed your presentation. Sort of going back to the bacteria species that just sort of got into your membrane reactors, do you find oftentimes that those species can convert your substrates more efficiently? And if so, have you, for example, tried to say use the reactors as a means to isolate them for degrading your specific substrates?
Sure. Enid, you have great insight. Congratulations. That's exactly what we do. Because the membrane biofilm reactor is an incredibly strong ecological selector. You can really control, in this case, oxygen, one of the primary electron acceptors. Of course, we were controlling the organic electron donor that we were putting in, plus you're growing it in a biofilm, which makes the community very stable. It stays in the reactor in a very stable way. So the MBFR turns out to be a really strong selector, and we use it in exactly that way as you described. And we do it for many, many things. Let's see, some of the things we're doing these days. We're very interested now in the reduction of metals, M-E-T-A-L-S, metals. They're very prominent in mining wastewater and ore processing, metals recycling, and we're very interested in using hydrogen. Now we're using hydrogen delivery, not oxygen delivery. So we're delivering a gaseous electron donor, hydrogen, and we're allowing these metals to be the electron acceptor. So, the respiration of oxidized metals into reduced metals is kind of specialized, and it provides quite a strong selective pressure. So yeah, so there's a case where we're very, very successful in selecting for bacteria that are particularly good at reducing one or several of these oxidized metals.
Huh, interesting. And I guess what are the challenges of, say, scaling up those type of reactors to say like treat mine waste or contaminated soils?
Yeah, okay, so let me, there's two-part answer here. I mean, first of all, the membrane biofilm reactor is a scaled up commercialized technology. So the hydrogen-based membrane biofilm reactor is now used in full scale for mainly for reduction of nitrate and perchlorate from contaminated groundwaters. So the technology is out there in full scale already. When you go to metals, and I'm going to address your metal question directly. When you go to reducing metals, the basic concept is the same, but the big difference is that when you reduce the metals, they precipitate in the biofilm, either as an elemental form or as a sulfide. And so now you're accumulating mineral solids in the biofilm, and this will increase the amount of biofilm mass that's produced, and it will put an emphasis, greater emphasis on managing the biofilm so that you don't get too much biofilm, you don't clog everything up, so you can't get the water through, for example. So those are the special features when you're dealing with metals.
Okay, very interesting. Thank you.
Well, thank you again, everybody, for attending our environmental engineering graduate seminar. And thank you again, Professor Rittmann, for presenting today. This was a real treat and really enjoyable. I think this is one of the things that you do really well, is to, one, find interesting problems, and two, to merge different tools, traditional and emerging tools in such an innovative way. So thanks again for presenting.
Great, my pleasure. Thanks for inviting me.
All
Hopefully
right,
we'll see each other in person someday soon.
that would be great.
Yes.
Thanks, everybody, and we'll see you next week. We'll be hearing from Detlef Knappe at North Carolina State University, and he'll be talking about his experience studying PFAS in North Carolina.
All right.
All right.
All right. Bye, everybody.